Assalamualaikum, welcome back to my video. We've done on the link reaction. Now let's move on the Krebs cycle. So Krebs cycle is the third process in cellular respiration. Let's revise what happened to the pyruvate. So pyruvate need to convert itself into acetyl coenzyme A before it can continue to the Krebs cycle during pyruvate oxidation. Now we have the acetyl coenzyme A. In Krebs cycle, there are 8 distinct steps and each step catalyzed by specific enzyme. So step number 1 is acetyl coenzyme A enter the cycle, and coenzyme A is stripped away from acetyl coenzyme A. So coenzyme A will be recycled by another process. So acetyl that contain 2 carbon atom combined with the oxaloacetate. Oxaloacetate has 4 carbon atoms. So 2 carbon atom plus 4 carbon atom forming 6 carbon atom named citrate, and this reaction catalyzed by citrate synthase. Second step is the isomerization of citrate into its isomer, which is the isocitrate. Isocitrate still have 6 carbon atom. So citrate is rearranged by removal and addition of water molecule forming isocitrate. An enzyme that catalyzes this reaction is aconitis. Step number 3 is the conversion of isocitrate into alpha-ketoglutarate. In this step, there is 2 processes involved. One is oxidation reduction process and the second is the decarboxylation. Okay, the first process is oxidation reduction, whereby isocitrate will be oxidized by losing electron and hydrogen ion. So NAD+ will be reduced by gaining electron and hydrogen ion from isocitrate. Next process is the decarboxylation. So isocitrate will lose 1 carboxyl group from 6 carbon atom forming 5 carbon atom, because decarboxylation means a removal of carboxyl group. So 5 carbon atom named as alpha-ketoglutarate. So this reaction catalyzed by isocitrate dehydrogenase. Step number 4 is another oxidation reduction and decarboxylation. So the first process is oxidation and reduction of alpha-ketoglutarate. So alpha-ketoglutarate oxidized by losing electron and hydrogen ion, while NAD+ will be reduced by gaining electrons and hydrogen ions from alpha-ketoglutarate forming NADH+. The second process is decarboxylation of alpha-ketoglutarate. So alpha-ketoglutarate contains 5 carbon atom. It removes 1 carbon dioxide forming 4 carbon product. Okay, so removal of 1 carboxyl group remove as carbon dioxide. And then coenzyme A will combine with the 4 carbon and forming product succinyl coenzyme A. Step number 5 is the formation of ATP via substrate level phosphorylation. In this step, coenzyme is displaced by inorganic phosphate, and then that inorganic phosphate will be transferred to guanosine diphosphate forming guanosine triphosphate. And then the next reaction is the inorganic phosphate will transfer to ADP, adenosine diphosphate, forming adenosine triphosphate, ATP. And the product is succinate, also contain 4 carbon atom. And this reaction mediated by succinyl coenzyme A synthetase enzyme. The next step, step number 6, is the oxidation of succinate to form fumarate. Fumarate has a 4 carbon atom. So succinate oxidized by losing electrons and hydrogen ion, while FAD reduced by gaining and receiving electrons and hydrogen ion from succinate forming FADH2, and this reaction catalyzed by succinate dehydrogenase. The next step is hydration of the fumarate forming malate. So fumarate is converted to malate by addition of water, and this reaction catalyzed by fumarase enzyme. And the last step is the formation of oxaloacetate from malate. So another oxidation process occurs here. So malate oxidized by losing electron and hydrogen ion, while another NAD plus will reduce gains and receiving hydrogen ion and electrons from malate, forming another NADH and regenerate the oxaloacetate. And this reaction is catalyzed by malate dehydrogenase. Now oxaloacetate is available for another reaction with another acetyl coenzyme A. Now let's summarize all the steps in Krebs cycle. So Krebs cycle happen in the matrix of mitochondrion. Each turn of the Krebs cycle will produce 3 NADH, 1 FADH2, and 2 carbon dioxide, and also 1 ATP. So for each glucose, since glucose will split into 2 pyruvate, and 2 pyruvate needs to convert itself into 2 acetyl coenzyme A, so the whole process forming 6 NADH, 2 FADH2, 4 carbon dioxide, and 2 ATP.